Nehs. She did her PhD at the University of Michigan, followed by a postdoc here in Boston at MIT and MGH, investigating the neural circuits that regulate sleep and are influenced by general anesthetics. She now runs a rodent neurometabolism lab in the anesthesia department here at MGH, studying the effects of metabolism and sleep on disrupted sleep with aging, postoperative delirium, Alzheimer's disease, and mental health. The title of her presentation today is Metabolic Interventions for Sleep, Delirium, Alzheimer's Disease, and Mental Health. Welcome, Dr. Nehs.
Great. Thank you for having me today.
Mhm.
Just
And it's our understanding that you're okay with recording your
Yes.
presentation today? Okay.
Yep.
All right. Take it away. Thank you.
All right. Let's see. All right. Can you all see my screen?
Looks great.
All right. Great. Well, thank you so much for the invitation to present my work to you all today. I'm really excited because I also have a shared interest in lifestyle strategies that prevent chronic diseases and maintain brain health with aging. My background, as you mentioned, is in more neural circuits that regulate sleep and are influenced by anesthesia. But I've always personally been interested in metabolism and sleep for both weight management and prevention of chronic diseases. And now I'm at a point where I'm fortunate to be able to connect my personal and professional interests to study the intersection of metabolism and sleep in health and disease. So the central focus of my lab is really this core idea that metabolism and sleep can be used as interventions to improve brain health. And I've applied this. There's many different areas of the brain that you could apply this to, but I have a background in sleep and anesthesia. And so for now, I've applied that to 4 different things, including the disrupted sleep with aging, postoperative delirium, Alzheimer's disease, and now most recently, mental health. So poor body metabolism influences brain metabolism, and the metabolic health in the US is getting worse and worse. As many of you know, a good measure of metabolic health is metabolic syndrome, and it's characterized as having 3 of the following criteria: obesity, blood pressure, high triglycerides, low HDL, or high insulin resistance. And this is a problem because it increases your risk of cardiovascular disease, diabetes, stroke, and other neurological complications. And remarkably, 1 in 3 adults have metabolic syndrome in the US, and over half of adults now are diabetic or pre-diabetic. And a proxy for metabolic syndrome is obesity. And if you look at this map from 1990 to 2021 of body mass index over 30, there is a significant increase in obesity in the last 30 years, from around 10 to 14% max in the 1990s up to now 45% in some regions of the US. And metabolic syndrome gets worse with age, and we have an aging population. And how does this relate to the brain? So all cells in the body need energy to generate ATP. And while the brain is only 2% of our body weight, it uses 20% of our energy. So with our brain's high energy demand, it needs this continuous supply of fuel to maintain consciousness. And interestingly, our brain is a bit like a hybrid car. It can run on glucose or ketones. And ketones generate more energy with less oxidative stress in fewer steps and have less metabolic waste than glucose. So what are the ketones? There are 3 main ones: acetone, acetoacetate, and beta hydroxybutyrate. They can be produced naturally from the liver during exercise, prolonged fasting, ketogenic diets, or now there's more and more ketone supplements that can be used. And many different dietary patterns can be used to generate ketones in the blood. So not only the brain, but the heart and skeletal muscle can also use ketones. And in addition to being a fuel source, they're potent potent signaling activity with decreasing oxidative stress and are anti-inflammatory. And if you've heard of ketoacidosis, that is in the range of 10 millimolar. And what we're talking about here is more in the range of 1 to 3. beginning nutritional ketosis starts around 0.5 millimolar. So we're an order of magnitude lower than what would be a problem in ketoacidosis. So what is the evolutionary significance of ketones? Why are they important? Well, historically, food was scarce and humans regularly went through periods of time where they had to fast, and the ability to store and use fat was advantageous. Our ancestors that survived were the ones that could use their own fat as fuel. This ketogenesis helped fuel the enlargement of the human brain during its evolution. And in today's food environment, where there's this overabundance of low quality food, the ability to store fat is less helpful, but our body's ability to use these ketones as fuel can still be advantageous. In fact, it's been known for over a century that ketogenic metabolic therapies are useful for epilepsy. And even in the 5th century BC, Hippocrates noted that the complete abstinence of food and drink was an effective cure for a man with epilepsy. In the 1920s, the ketogenic diet was developed at Johns Hopkins as a fasting mimicking diet for pediatric epilepsy. And what's remarkable about this is that the diet actually decreases seizures in epilepsy with many different etiologies. And for some people, it actually cures it completely. In children that have epilepsy and are not able to have treatments with drugs that work, they can go onto the diet for 2 to 5 years and then come off of the diet and remain seizure-free for the rest of their lives. So with age, there's also changes in brain metabolism, including mitochondrial dysfunction, decreased glucose uptake and insulin resistance. There's increased oxidative stress and inflammation, and the brain's ability to use this glucose declines, but the ability of the brain to use ketones remains. And I'll just show an example of that from a PET imaging study where glucose and ketone utilization is shown in a healthy control, somebody with mild cognitive impairment and Alzheimer's disease. And here you can see the PET imaging for glucose where it's very robust in the control and it declines as their cognitive impairment increases. And then there's a tracer for acetoacetate, one of the ketone bodies, and you can see that even in the older brain with Alzheimer's disease, there's still the ability to use ketones as fuel. So this demonstrates that the deterioration in brain energy metabolism with mild cognitive impairment and Alzheimer's disease is specific to glucose. So we can use this older brain's ability to use ketones as a potential therapeutic. And there's many different problems in the brain that have metabolic dysregulation, including sleep disruption, postoperative delirium, the neurodegenerative diseases like Alzheimer's disease and Parkinson's disease, mental health, including anxiety, bipolar disorder, schizophrenia, and major depression, and epilepsy, as we just talked about, but also autism and many cancers, including glioblastoma. And my lab focuses on the first 4 of these, looking at this disrupted sleep with aging, how decreased metabolic dysfunction in the older brain could lead to postoperative delirium and be used as a prevention treatment for delirium, neurodegenerative diseases like Alzheimer's disease, and then most recently adding in mental health. So first I'll go through disrupted sleep with aging. So in the aging process, both non-REM and REM sleep decline, and especially deep slow wave sleep. Sleep becomes more fragmented, and there's changes in circadian rhythms as well, with phase advancement, decreased ability to adjust to phase shifts, and reduced amplitude of circadian rhythms. So this disrupted sleep also alters glucose metabolism and insulin resistance, and also appetite hormones. So what links this disrupted sleep to metabolism? Well, my hypothesis is that this is likely through, at least in part, oxidative stress. So oxidative stress is an imbalance of the reactive oxygen species generated by metabolic processes and antioxidant responses. So oxidative stress can damage lipids, proteins, and DNA, and is associated with aging. There's a bidirectional relationship between sleep and oxidative stress, where sleep loss increases reactive oxygen species, and the increase in oxidative stress can trigger sleep. So ketones improve mitochondrial function and significantly reduce oxidative stress, increase antioxidants and scavenge free radicals. So these two mechanisms of modulating sleep and modulating metabolism through ketones can modulate oxidative stress. And we're going to be able to do that in the context of the aging brain and sleep. So there's some evidence that this might be true. In an animal model, there was an injection of the ketone acetoacetate into the ventricle, and it increased slow wave activity, shown here, in the first few hours after sleep. So this suggests that ketones could be a promising way to improve sleep quality, because again, this disrupted slow wave activity is one of the key features of aging. Now, in addition, in humans, I've collaborated with a company called Virta Health that is using the ketogenic diet to reverse type 2 diabetes. And their whole goal is to deprescribe medications. And so this is a study where they looked at their protocol in type 2 diabetic patients, people with pre-diabetes, and then people with type 2 diabetes that just followed the standard of care. And you can see here, we used a Pittsburgh Sleep Quality Index questionnaire to look at the quality of their sleep with these different interventions. You can see that their sleep improved overall with the intervention, the ketogenic diet intervention, as their diabetes symptoms improved. Their subjective sleep quality and their sleep disturbance and daytime dysfunction all improved compared to the usual care group. So how does metabolism change across sleep and wakefulness? And so this is a nice summary diagram that shows what's happening in the blood versus in the brain across sleep, primarily non-REM sleep, compared to wakefulness. So in the blood, when you're asleep, your glucose goes down because you're not eating, while your body shifts to free fatty acids increasing and ketone bodies increasing over time. Interestingly, in the brain, your blood glucose actually goes up during sleep, which is the opposite of what's going on in the body. And so also your antioxidant capacity increases, and then your glucose metabolic rate goes down, and your oxygen metabolic rate goes down a little bit, but not quite as much, suggesting that there's a shift in energy from utilization from glucose to fatty acids. So in order to investigate this further, in my lab, we used a biosensor that is specific for either glucose or lactate. And so you can place this in different parts of the rodent brain. We looked at the prefrontal cortex, and it can detect the analyte of interest, in this case, lactate, with an enzyme that turns into hydrogen peroxide in the presence of lactate and causes a potential on a platinum electrode. And then you can measure in real time the second-to-second differences in glucose or lactate in the brain across behavioral states. So our experiment design includes inserting the probe. You have to let it acclimate for a little while after you put it in there. And then you can record both sleep and wake cycles, as well as anesthesia, to look at the differences of what's happening between glucose and lactate during sleep versus during anesthesia and recovery times. So first I'll go through the sleep data. So at the top here, we have a hypnogram showing when the animal is awake versus non-REM sleep. And then in part C here, we have the glucose in blue and the lactate in red. And you can see there's this biphasic relationship that usually they're kind of anti-phase with each other. And at the bottom here is the spectrogram of the EEG. So you can see the different oscillations that are present in the different states. If you look at this period of wakefulness here, you can see that when the animal wakes up, lactate increases and glucose decreases. And when the animal goes to sleep, for example, here, the lactate decreases and the glucose increases. And that could be due to a shift in the utilization of away from glucose towards other fuels, and also possibly lymphatic clearance of lactate or utilization of lactate during non-REM sleep. So if you look at what's happening during anesthesia, we see a similar pattern, but a larger magnitude. So we used the sevoflurane as our anesthetic and ramped it up gradually over time, so you could see an induction period. And here you can see that there's this metabolic switch of this lactate and glucose right around the time when there's high delta power and deeper anesthesia. And then that's similar to non-REM sleep. When the animal wakes up, similar to wake, lactate goes up and glucose goes down. Now, if we compare between sleep and between anesthesia, what's happening, you can see here that these little bumps are when, this is in lactate, when when wake occurs. And then over here in sevoflurane, this is on the same scale. You can see that both glucose and lactate increase dramatically during sevoflurane anesthesia, and that their pattern remains similar. Here is the similar to deep non-REM sleep, and here is when they wake up. This is just a single animal. If we look at the group data, you can see that there's a big difference in the amount of lactate between wake and this induction period and emergence. There's also a larger amount of lactate during deep anesthesia compared to non-REM sleep. And then for glucose, you can see that the range, this is the standard deviation. So there's a bigger difference in the glucose levels during sevoflurane where it fluctuates. Also with lactate in sevoflurane compared to sleep and wakefulness. So with that, I'll move on to the metabolic neuroprotection to prevent delirium. So postoperative delirium is an acute syndrome characterized by inattention, disorganized thinking, altered levels of consciousness, and it occurs more often in older people. So the consequences of postoperative delirium include prolonged ICU stay, long-term neurocognitive deficits, and increased mortality. So the pathogenesis of delirium is not completely understood, but aging, inflammation, vascular dysfunction, disrupted brain metabolism, sleep disruption, neurotransmitter imbalance, and impaired network connectivity are thought to contribute. So one way of looking at the things that influence postoperative delirium is looking at predisposing factors. So the same age older person could be very healthy or they could be very frail. So the person coming in has a differential risk factor just to start with, from the inflammation level, from the vascular level, from their oxidative stress, and then also like neuroendocrine and neuroanatomical abnormalities. So these things influence whether the acute stressors of surgery and anesthesia are going to lead to delirium. And so with delirium, there's more inflammation, microglial activation, physical stress, there's changes in neurotransmitters, and the medications. And the consequence, as I mentioned, is that there's poor health-related outcomes. There's disability and hospitalization, and ultimately could be death. So what are factors that contribute to this that I'm particularly interested in? Again, I like to look at sleep and metabolism as ways that modulate these things. So poor sleep quality and delirium are common, and they often coexist. And sleep quality declines with age, while the probability of delirium increases. So disrupted sleep before surgery can contribute to an increased risk of delirium, as well as disrupted sleep after surgery from many different things, including the drugs and lights and hospital interactions in the ICU that can increase this risk of delirium. What about metabolic contributions to delirium? So recently, metabolic syndrome had been shown to be a significantly increased risk factor for developing postoperative delirium. And in addition, acute surgical inflammation disrupts energy metabolism in older people, increasing their chance of having delirium. Another thing that connects metabolism to delirium is this study that looked at the lipid metabolism in different people and showed there was altered lipid metabolism in the people that were likely to develop delirium. In addition, there's low baseline antioxidant capacity can be independently associated with postoperative delirium, suggesting that oxidative stress is involved in this postoperative delirium phenotype. So I want to introduce the concept of burst suppression here. So during deep anesthesia, during hypothermia, sometimes in coma, there's a pattern in the brain where the brain goes to this periods of bursts and then quiet periods and then another burst. So this is called burst suppression, as opposed to what we normally see when they're awake, which is some form of continuous activity like that. So in the operating room, it's been observed that how much time the patient spends in burst suppression correlates with the likelihood of them developing delirium. However, when people have gone in and adjusted the drug levels to try and have less burst suppression, there's been mixed results as to whether that's been an effective strategy for decreasing delirium. And one reason that might be is that just decreasing the drug is not doing anything to fundamentally improve whatever the underlying dysfunction in the brain is that is generating that pattern to start with. So there's a couple of different theories of burst suppression, but one involves that burst suppression occurs when ATP runs low due to this intersection of neuronal dynamics and brain metabolism. And so it's the brain's way of handling low energy, low ATP levels. So we hypothesized that ketones could be neuroprotective during anesthesia by providing this alternate energy source, decreasing oxidative stress and inflammation. And so we had animals on either the ketogenic diet or a standard diet and looked at their level of burst suppression at the same level of sevoflurane anesthesia. So at the top here, these are animals on a standard diet. The black bar indicates when sevoflurane is present, and this is the spectrogram of the EEG. And here the BSR is the burst suppression ratio. So you can see there's quite a bit of burst suppression in an animal at this level of sevoflurane on a standard diet. When the animal is on a ketogenic diet, they're at the same level of sevoflurane anesthesia, but there's much less burst suppression. And this is one example, and if we quantify that, you can see that there is quite a significant decrease between the standard diet and the ketogenic diet in their burst suppression ratio. Interestingly, it also took longer for the animals on the ketogenic diet to become anesthetized, as though they were resistant to becoming anesthetized. The time to return of movement after anesthesia did not change, and that's possibly due to the fact that the process of coming out of anesthesia is due to metabolism of the drug, which we're not influencing here. So these data suggest that this presence of ketones could be neuroprotective and have the potential to decrease postoperative delirium. And so we're now doing this, adding cognitive tests to the rodent experiments. And the nice thing about ketones is that they're readily available to translate to humans. So we're working on a study to be able to test this in humans in the ICU here at MGH as well. So just a little bit more detail about burst suppression. There's kind of two prevailing theories about why it exists. I mentioned this first one already. It's kind of the hypometabolism theory, that burst suppression reflects a decrease in neural metabolism and a deficit in ATP. Well, there's another theory that says that it's a hyperexcitable state, and that burst suppression results from cortical excitability arising from an imbalance of excitation and inhibition. And so our data lends a little bit of clues as to support for actually each one of these theories contributing to the role of burst suppression. So this is the over time data from what I had just shown you of animals on a standard diet in this pink color and on the ketogenic diet with their confidence intervals here in blue. And then the next figure in purple is the difference, the confidence intervals of the difference. And you can see that they're significantly different for the majority of the time. Well, in epilepsy literature, there is evidence that DPCPX, which is an adenosine A1 receptor antagonist, that it can reverse the ketogenic diet's suppression of seizures. And so we thought perhaps that this ketogenic diet is working through this adenosine receptor as well for burst suppression. So we gave the adenosine A1 receptor antagonist, DPCPX, to the animals, and we were able to show that it increased the amount of burst suppression that had previously been suppressed by the ketogenic diet, despite these animals still having the still being on the ketogenic diet, and not fully, but at least partially. So here's a little diagram from the epilepsy world that helps to explain why this might be happening. So ketone bodies increase ATP, ATP increases extracellularly and increases adenosine, which binds to the adenosine A1 receptor and activates ATP-dependent potassium channels, which leads to hyperpolarization and stabilizing of the neural network. So you have an anti-epileptic effect in that case, and in our case, you get less burst suppression. So just to summarize that, our data supports both theories that ketones provide additional fuel to generate ATP, so the brain doesn't run out of energy as quickly. And our adenosine A1 receptor antagonist data shows that ketones stabilize neural excitability, similar to epilepsy, likely through the ATP-dependent potassium channels. So with that, I'll just briefly go through how we're moving on to look at both Alzheimer's disease and mental health studies. So as many of you know, the glucose hypometabolism and sleep disruption are early indicators of Alzheimer's disease. And there's been many both animal and human studies looking at ketones, and they've shown promising data of improving cognitive function in both animals and humans. So we're really interested in characterizing this energy dysfunction in Alzheimer's disease down to the mitochondrial level with various imaging techniques, and looking at how disrupted or enhanced sleep can affect Alzheimer's disease progression. And this is an ongoing area of research in the lab. Next, we're really getting interested in metabolic and sleep interventions to treat psychiatric disorders. So bipolar disorder and schizophrenia are both serious psychiatric conditions that cause a significant reduction in the quality of life and shortened life expectancy. While their exact etiology remains unclear, there's increasing evidence supporting the role of oxidative stress and redox dysregulation as underlying mechanisms. In addition, sleep is a critical regulator of this metabolic homeostasis and oxidative stress, and disrupted sleep and circadian rhythms can contribute to the onset and the progression of bipolar disorder and schizophrenia. So recently, there's been some case studies and now some clinical trials that have investigated the role of the ketogenic diet to improve bipolar disorder and schizophrenia symptoms. And so therefore, we're really interested in this intersection of sleep, metabolism, and mental health, and how the three of those things are intricately connected. So we're working on modulating sleep and measuring changes in mood symptoms in these animal models of bipolar disorder and schizophrenia. And another nice aspect of this is that there's ongoing human trials as well that we can collaborate with them to look at how sleep and circadian rhythms are changing in human patients that are undergoing the ketogenic diet to improve their mood symptoms. And for more information, there's an excellent book on this subject called Brain Energy by Chris Palmer, who's here at McLean Hospital, that really dives into the details of how this theory works in mental health and the opportunities there to be able to use it to improve the field of mental health. So my summary and future directions: if we can improve our understanding of brain metabolism across these conditions, there's potential for broad impact, since many diseases have these components of metabolic dysregulation. And as I've mentioned, it's readily translatable to humans. So we're actively working on ketone interventions for human delirium and these psychiatric disorders. And these findings could lead to new metabolic and sleep therapies for aging, disrupted sleep with aging, postoperative delirium, slowing the progression of Alzheimer's disease, and improving mental health. So what are strategies to improve the health span and not just the lifespan? I'm personally and professionally interested in this. And as you know, lifespan is the total length of one's life, whereas health span is the period of time where you're free from chronic diseases. And so I'm really interested in, is the health span predetermined, or what can you do to influence this? What are modifiable lifestyle factors that you can control to influence your health span? Sort of similar to Dr. Tanzi's SHIELD protocol. These are things that I've identified that are helpful for improving your brain health and preventing chronic diseases. So obviously things that I care about are sleep. So having good quality sleep, good sleep hygiene, going to bed at the same time, waking up at the same time each day, and making sure you get sufficient sleep, avoiding stimulants and alcohol use close to bedtime. And then another big important thing is quality nutrition, right? And that can range from a whole bunch of different things, but I think some key points are eliminating the ultra-processed foods that are full of these seed oils and added sugars and refined grains, decreasing the amount of omega-6 fatty acids and increasing omega-3 fatty acids, and then just focusing on whole nutrient-dense foods so you get your micronutrients. And then fasting is a really powerful tool. It's one of the world's oldest cures. So periodically fasting or integrating fasting into your lifestyle can give you a lot more flexibility on what it is you eat when you eat. So another important aspect that some of you I know study, moving your body, including walking, aerobic exercise, weight training, integrating activity into your daily life is really important. Getting outside. So natural sunlight exposure first thing in the morning helps set your circadian rhythms, and some midday sun for vitamin D. And in Boston, you can't make vitamin D from November to March, so maybe consider a supplement. Managing stress through meditation and time management strategies. Avoiding environmental toxins, some of these endocrine disruptors like environmental estrogens or glyphosate, mold, and these others. So a thing that's a little bit harder to study in the rodents, but very important for humans, is building strong relationships and social support. We tell our children, choose your life partner wisely. And then the Japanese concept of ikigai, finding your purpose in life, or finding one's passion that gives you value and joy, and practicing gratitude. So these things do many things, including optimize our circadian rhythms and sleep and keeping our mitochondria healthy. So with that, I'd like to thank my lab members here. You can see we're also in Charlestown, have a number of postdocs and some summer students that have worked on the projects, and my funding sources as well. Thank you.
Thank you very much. Let's see. Do we have any questions or comments? Go ahead, Jonathan.
Christa, congratulations. That was a terrific talk, and it's wonderful to see so much high quality science kind of converging on a vision for prevention. We've been talking with colleagues of yours in anesthesia for a while about the opportunity for delirium prevention as a way of probably reducing the risk of dementia at a broad level. And so what I was wondering is do you see a future where we might be encouraging people to adopt a certain diet a couple of days, a week or so before their elective anesthesias as one approach?
I think so. I think that would be helpful. And in the absence of being able to change older person's acute diet, there are now these tools of ketone esters or MCT oil where you can increase at least the ketones during the surgery or immediately after surgery to help have that additional protection without having to necessarily change one's diet drastically in that time leading up to the surgery. Ideally, everybody could, but realistically, that might not happen. And then there are these handy tools now where you don't have to change your diet, but you can get this increased level of ketones.
And are people thinking about clinical trials in this space?
So we're thinking about that here. And then I have some collaborators at Duke and the Buck Institute for Aging that are interested in this as well, that we're hoping to put together a clinical trial for.
Fantastic.
Christa, hey, it's Rudy here. Great talk. You're really speaking our language, and I love the recommendations at the end that resonate so well with Shield. I think adding going outside is great, and taking a D3 supplement is a great idea as well. I had a specific question about the anesthesia because our group had published several papers about isoflurane in particular versus desflurane or sevoflurane, inducing A beta generation. And I think this is Zung Shi's work from anesthesia, who when he was in my lab.
Right.
And the order of severity in terms of inducing apoptosis and A beta, et cetera, was isoflurane was the worst. sevoflurane was in the middle and desflurane was the best, but I know des is expensive and it's also not environmentally a great gas. Have you looked at the differences with different gases just to see what their interaction with the ketones, given that information?
Yeah, so we are just starting to do that now. We started with sevoflurane, as that's the most clinically used at the moment, but we would love to see that with the different anesthetics, how that changes, and then look at how that we can look at how that affects like Alzheimer's pathology too. That's in our future studies.
So isoflurane is not as commonly used anymore?
Not as commonly used in the OR, no.
Okay, and I guess desflurane is just too expensive, and I guess there's issues
A
with
little
how
bit,
it's
but
made. Yeah,
right.
Yeah, gotcha. Okay. Well, I'd love to hear those results when you have them. Thank you.
Yeah.
And again, great talk. Really enjoyed
Thank
it.
you.
Dr. Rand?
Yeah. Hi, hi, Christa. Very nice talk. I'm really glad to kind of see all the work, how it has come together. I have a question. I know I was aware about the work on epilepsy and the ketogenic diet. Are there, is it always, it's shifting kind of the level of excitation that you can have. Is this always a good thing? I mean, for epilepsy and I think the delirium, but are there other areas of the brain where you don't want to have that increased threshold, or is it affecting different brain circuits in a different way? If you think about, maybe, we're mostly interested in cognitive function and memory function.
Right. So it's interesting because it's neurostabilizing and it both prevents the aberrant activity of seizures and seemingly burst suppression, but it also improves cognitive function in Alzheimer's disease. And people will take ketones or have higher ketones from the ketogenic diet and then see improved focus and improved clarity. So it's an interesting phenomenon where it both kind of boosts your daytime function and maybe your nighttime quality of sleep, and kind of keeps the brain at a stable level of not having hyperexcitability.
So maybe this is kind of, you could think about it as like the noise in the background kind of thing that happens in Alzheimer's disease, that you have more leaky circuits that are responding maybe to stimuli that they should not, and that a young healthy patient can actively suppress, and that's why you can still focus. But if you go into Alzheimer's disease, this suppression or the stabilizing function is just decreasing in all different kinds of areas in the brain.
Yeah.
Okay. Very interesting. And then, of course, I have to ask, what does exercise do to the ketone levels in the brain? I mean, I might know the answer, but I actually
well,
since we
depending on how intense and how much you've eaten of carbs beforehand, but it's actually very interesting. So Jeff Volek at Ohio State has done a lot of work with ketones and endurance exercise. There's been a number of like 100-mile races won by people who are fueling on ketones instead of glucose. And that's because you don't run out, you never hit the bonk point. You never run out of the ketones. Even a lean person still has a lot of fat on their body that they can use as fuel if they're keto adapted. So people, when they start to go on the ketogenic diet, often have maybe some temporary decreases in performance, but then usually as they adapt, it comes back. And so I don't think people have measured brain ketone levels during these intense exercise studies, but that's something that, we're talking to people about exercise and being able to see how that influences ketones. So, or we could collaborate too.
I just want to say, like, if you do it, not in humans, if you want to do it in mice, you should just come over. We have plenty of brains.
Right. Yeah.
So maybe just a follow-up question to that. You have this interesting rodent model, and you mentioned medium chain triglycerides. I'm wondering, have you attempted to try different chain length medium chain triglycerides, so C8, C10, C11?
Right. I think C8 is the most ketogenic and has the least digestive distress. So kind of the supplements out there that are primarily C8 are kind of the most optimal, but C8, C10 are both very effective at generating ketones. And, like coconut oil has some, but it's a bit more mixed of different fatty acid profiles. So there's a number of companies making kind of purified C8 as probably the go-to MCT oil.
Okay. Interesting. And what about the ketones? So different ketones. Have you tried the different types of ketones in your?
Yeah, so there's been ketone salts on the market for a long time, and they come in all different flavors and varying levels of potency. But in the recent years, there's been a number of developments on ketone esters, which are nice because they increase ketones a little bit more slowly and maintain the ketones at a higher level for a longer period of time. The downside is the original ones tasted like jet fuel, so it was pretty non-palatable, but there's a number of companies coming up with better formulations to try and improve the palatability of these ketone esters that have potential for like these therapeutic benefits. So like people with epilepsy could supplement with some of these ketone esters or the MCTs and not have to be quite so strict on their diet.
Interesting. And what about the delivery across the blood-brain barrier? I mean, is there variability based on the transport mechanisms or breakdown in these in terms of, I guess, in living subjects where you have a delirium? Is there variability at the delivery of the ketone?
Yeah, ketones are taken up readily by MCT transporters, similar to lactate. They, there are polymorphisms in the MCT receptors, and so I don't think anybody's actually looked at that in the context of delirium, but that's something we could do. Yeah.
Interesting. Very interesting talk. Thank you very much. Are there any more questions? Okay. Well, thank you very much for the presentation today. And thank you, Dr. Rosen, for the invite of the very interesting, talented faculty member close by.
Thank you. Yeah, thank you
Nice
for
to meet
having
you.
me. It's
Yeah.
nice to meet you all.
Great job.
Okay.
Thank
Thank
you.
you, everybody. Thank you for attending. Take care.